Hello and welcome to MedMind Spectrum. In this video, you will learn about the fate of the pyruvate. Like the word itself, we want to understand what happens to pyruvate at the end of glycolysis. Remember, pyruvate was one of our end products of glycolysis. And if you haven't watched the glycolysis video yet, check in the description below this video and first watch that video so that you can keep pace with this one so that we can easily understand what happens to pyruvate at the end of glycolysis. Now remember, from our glycolysis, we had two pyruvate molecules produced as end products, and these were 3 carbon compounds. We had 2 NADH molecules. We also had 4 gross ATP molecules and 2 net ATP molecules. But in this particular video, we want to understand what happens to pyruvate and then what happens to NADH molecules. Of course, what happens to ATP? ATP is used for energy demanding processes, say for example muscle contraction and many more. In this case, we want to understand the pyruvate we have formed, is it a waste product, or if it is useful, then how is it useful? Now there are basically 4 pathways or 4 routes we can have pyruvate take at the end of glycolysis. One of them, we can have reversible conversion to lactate. We can convert pyruvate to lactate, and this reaction is reversible. It means that once we need pyruvate back, we can have lactate as one of the sources of this pyruvate. This becomes very important in processes like gluconeogenesis, where we want to make carbohydrates like glucose from other non-carbohydrate sources. So lactate becomes very important. But in this case, we are interested in learning how is pyruvate converted into lactate. This is a reduction reaction. We simply use NADH molecules to reduce pyruvate to lactate, and in this case we shall produce NAD positive because we are reducing. And this is really important because NAD positive that we have produced is going to be used again in the glycolytic reactions, which are basically generating ATP. So it makes us provide NAD positive molecules that are going to be eventually recycled in the process of glycolysis so that we can make more and more ATP. This is very important in anaerobic conditions and in cells like red blood cells that entirely depend on glycolysis for their glycolysis to continue operating using NADP positive that is regenerated in this process. Now, we can also have irreversible conversion to acetyl-CoA. It means that once we convert pyruvate to acetyl-CoA, we cannot have it brought back by the same enzyme. In this case, we have to use other intermediates or even not possible at all. So it means this step is highly regulated, because once we spend our energy to form acetyl-CoA and we need pyruvate back, we cannot have it. That means we should only form acetyl-CoA if need be. And this reaction is particularly important because acetyl-CoA is one of the molecules we use in the tricarboxylic cycle, which forms more ATP and more NADH molecules. This usually occurs in presence of oxygen because that's why we have the Krebs cycle operating, so that we can have more energy generated. The third way can be reversible conversion to alanine. Now alanine is one of the amino acids that we know about that are non-polar in form. So pyruvate can be transaminated to amino acid called alanine, and in transamination we basically transfer an amine group to pyruvate and then we eventually form alanine. But this reaction is as well reversible. That means if we need pyruvate, one of the sources of pyruvate can also be alanine, because we just instead now deaminate it and then we have pyruvate back. The last pathway or direction pyruvate can take is to be converted into oxaloacetate. And once it's converted to oxaloacetate, this is very important for replenishing the tricarboxylic cycle, because the Krebs cycle is where we have known oxaloacetate very much useful as the first molecule of the reaction. The details of the Krebs cycle are also in another video which you can check out and enjoy it as well. So in this particular case, let's look at the details of how this process occurs. I want to remind you, from glycolysis, we had our glucose enter the cell. We converted it into fructose 1,6-bisphosphate, and then cleaved this fructose 1,6-bisphosphate into 2 molecules, dihydroxyacetone phosphate and then glyceraldehyde 3-phosphate. And then after that, we isomerize DHAP to GA3P, because we said GA3P can only proceed with the next reactions. Dihydroxyacetone phosphate cannot. That means we have to isomerize it to the next Ga3P, and then we have glycolytic reactions proceed. And then after that, we have this one converted to pyruvate through a series of very many steps, but in this case, we are interested in the pyruvate. Now, we want to see what happens to pyruvate at the end of glycolysis. So we've said one of the pathways we can convert it into lactate, and once we get it to lactate, the enzyme in this case is called lactate dehydrogenase enzyme. And in case it is a dehydrogenase enzyme, you begin to click, oh, we may have NADH or NAD positive somewhere. And in this case, we are using NADH because the dehydrogenase activity is for lactate. That means we use NAD positive if we are coming from lactate to pyruvate. But in this case, we are turning pyruvate into lactate. That means we shall use NADH molecules and then convert them into NAD positive. This is important because remember, in the reactions of glycolysis, NAD positive is important somewhere in this step of changing Ga3P to 1,3-bisphosphoglycerate. So once we form NAD positive, we can bring it back to glycolysis and we allow glycolysis to proceed again and again. The second reaction is converting pyruvate into acetyl-CoA, and this reaction occurs in the mitochondria. Now you can see pyruvate has 3 carbons, whereas acetyl-CoA has only 2 carbons. What happened to the other carbon? It was lost. And in most cases, we lose carbon in form of carbon dioxide. That means we lose it as a carbon dioxide, and this carbon dioxide gets off this reaction. And in this case, this enzyme, people usually want to confuse it as pyruvate decarboxylase. But in this case, it is not a decarboxylase, but instead pyruvate dehydrogenase. Now, this enzyme is very important and highly controlled. Remember, this is a one-step forward reaction and it cannot be reversed. So this step is highly regulated. But this enzyme in particular uses a very important cofactor derived from vitamin B1, which is also known as thiamine, and this particular coenzyme is called thiamine pyrophosphate, which has been abbreviated as TPP, thiamine pyrophosphate, derived from vitamin B1. The details of this reaction and the control of pyruvate dehydrogenase are discussed in a video titled Control of the Transition State, which you can check out and see the beauty of this enzyme, how it works with its reactions of changing pyruvate into acetyl-CoA through a series of steps. But in this case, we shall not go into the detail. Like I said, it has a dehydrogenase activity. In this case, we shall utilize NAD positive and generate NADH molecules because we are dehydrogenating pyruvate. We are removing hydrogen atoms from pyruvate and we are using them to make NADH, which can later be used to synthesize ATP from the electron transport chain. The third pathway is to convert pyruvate into oxaloacetate. Now, oxaloacetate is one of the very important molecules that participate in the Krebs cycle. But the unique thing with oxaloacetate is that it is a 4-carbon compound, whereas pyruvate is a 3-carbon compound. How do we have another carbon get added to pyruvate and then we end up with 4 carbons? Think about carbon dioxide. So in this particular reaction, we add carbon dioxide to pyruvate and we have oxaloacetate. And this enzyme in this carboxylation reaction is called pyruvate carboxylase. And I think this makes a lot of sense because we are carboxylating a pyruvate molecule to form oxaloacetate. So the enzyme is called pyruvate carboxylase. Now this enzyme uses a very important cofactor derived from vitamin B7, and this is called biocytin. So this enzyme uses a cofactor derived from vitamin B7. You can see that this whole bunch of enzymes requires certain cofactors derived from vitamins. That means in case we suffer deficiency from vitamins, we may have some of these processes crippled because their enzymes may not proceed or work efficiently as they are supposed to be. Now, this particular reaction also demands energy in form of ATP. And ATP gets used up and then we generate ADP in the process and an inorganic phosphate. The fourth pathway or direction of pyruvate, it can get transaminated to alanine. And the enzyme involved in this case is alanine aminotransferase enzyme. It is basically important in transferring an amine group to pyruvate so as we can form alanine. Now this enzyme, a transaminase, uses another cofactor called pyridoxal This one is derived from pyridoxine vitamin B6, its other name. So We have seen we need vitamin B1 for thymine pyrophosphate, for pyruvate dehydrogenase, vitamin B7 for pyruvate carboxylase, and vitamin B6 for alanine aminotransferase. Now, we have looked at the fate of pyruvate. Let's now look at the fate of NADH. Remember, we said NADH cannot cross the mitochondrial membrane. And what do we do to transfer these electrons to the mitochondria? We have what we call shuttle systems. And in these shuttle systems, we produce what we call reducing equivalents. Remember, NADH molecules are reducing, or they have reducing power because they carry hydrogen atoms. So what happens in these shuttle systems is that we use NADH to reduce other molecules to form other reductive molecules which can enter the mitochondria. So once these enter the mitochondria, they have the reducing power which they inherited, I can say, from NADH molecules. One of these shuttle systems is the glycerophosphate shuttle. And in this case, we use DHAP in the cytoplasm, and then we form what we call glycerol 3-phosphate. This glycerol 3-phosphate is the reducing equivalent. Now, in this particular reaction, we use an enzyme called glycerol 3-phosphate dehydrogenase enzyme. Now, we also have another shuttle system called the malate aspartate shuttle. This one uses oxaloacetate in the cytoplasm, and we convert it into malate. Now, it is malate that is the reducing equivalent. So it is malate that carries these hydrogen atoms into the mitochondria, and in this case we use malate dehydrogenase enzyme. Let's look at the details of this process in the next slide. Now, what happens, remember from our glycolysis, we have glucose being converted into pyruvate, and in this case we generate NADH molecules. But we've said these NADH molecules cannot cross the mitochondria to enter into the mitochondrial matrix. So what happens is that we have dihydroxyacetone phosphate in the cytoplasm. This is a ketone, remember. So what happens is that we convert it into glycerol 3-phosphate. Now, from a dihydroxyacetone, which is a ketone, to an alcohol form, which is glycerol, it is basically a reduction reaction. Who provided this reduction power? It is basically NADH molecules. And in this case, once they do that, they get converted into NAD positive. Where have the hydrogen atoms gone? We have utilized them in reducing DHAP to form glycerol 3-phosphate. Now, glycerol 3-phosphate is the reducing equivalent. And now this one can easily enter the mitochondria, and then we make ATP via the electron transport chain. In this particular reaction, the enzyme involved is called cytosolic glycerol 3-phosphate dehydrogenase. Because it is a dehydrogenase of glycerol 3-phosphate, once it is acting in this other direction from glycerol 3-phosphate to DHAP, it will be removing hydrogen atoms from glycerol 3-phosphate, but in this case it is acting the opposite. That's why we are having NADH molecules being converted into NAD positive. Now we have formed the glycerol 3-phosphate, which can now enter the mitochondria, and once they are in the mitochondria, it gets converted back into DHAP, and it is going to release the hydrogen atoms that it previously acquired in reduction of DHAP. And guess who is going to accept these hydrogen atoms? It is none other than another molecule called FAD positive. But this reaction is catalyzed by the same enzyme, glycerol 3-phosphate dehydrogenase, and by this one, in this case, it is from the mitochondria and is called mitochondrial glycerol 3-phosphate dehydrogenase. We've said in this particular reaction, now the hydrogen atoms are going to be transferred back to another molecule, and this is called FAD positive. So once FAD positive accepts these electrons provided in form of hydride ions, we then form FADH, and now FADH can proceed to the electron transport chain, and we form our ATP. So basically, in this glycerol phosphate shuttle system, we have DHAP converted into glycerol 3-phosphate, and then glycerol 3-phosphate enters the mitochondrial membrane, specifically the inner one, and then converted back to DHAP, and we release those hydrogen atoms that were previously provided by NADH molecules. Let's now look at the malate aspartate shuttle. Now, in this particular shuttle, we are using malate, but we shall see how aspartate eventually comes in. Remember, from glycolysis, we have glucose getting converted into pyruvate and release NADH. But our problem is getting hydrogen atoms that are on ADH, putting them into the inner mitochondrial membrane so that we can have them utilized in the electron transport chain to produce more and more ATP. Remember, in the malate aspartate shuttle, we said we utilize oxaloacetate present in the cytosol. So this oxaloacetate, a 4-carbon compound, gets reduced to malate. Who has provided the reducing power? It is basically NADH, which gets converted into NAD positive. Now, once it has been converted to NAD positive, the malate we have formed enters the mitochondria. But the enzyme involved in this reduction of oxaloacetate to malate is called malate dehydrogenase. This one is specifically located in the cytosol. Now, the malate we have formed will now enter the mitochondria, and once in the mitochondria, it gets converted back into oxaloacetate. And of course, if it gets back into oxaloacetate, it must release the hydrogen atoms that it had acquired. And we shall have now NAD positive pick up these hydrogen atoms and then form NADH molecules. You can see our NADH is now safe in the mitochondria, and there it can proceed to the mitochondrial membrane, specifically the inner one, and then we make more and more ATP from the electron transport chain. But remember, if this is to proceed, we must have oxaloacetate being replenished over and over again, because if it enters and it cannot return to the cytoplasm, we shall have an imbalance of this reaction. So what happens to oxaloacetate after it has been formed inside the mitochondria? We convert oxaloacetate into aspartate. Because oxaloacetate cannot move across the mitochondrial membrane, that's why we need to convert it into aspartate. Now, it is aspartate that can get transported into the cytoplasm, and once it is in the cytoplasm, it can do a lot of things, which we shall look at in a second. Now, this particular reaction where we convert oxaloacetate into aspartate, it is a transamination reaction. Remember, oxaloacetate is one of the molecules or intermediates of Krebs cycle, but aspartate is an amino acid. Now, it means that we transferred an amino group to oxaloacetate to aspartate. Now, which molecule provides this amino group? It is another amino acid called glutamate. Now, glutamate, in this case, provides this amino group, which we add to oxaloacetate and then form aspartate. And in this case, once it does so, it gets converted to what we call alpha-ketoglutarate. Now, the enzyme involved in this reaction is called GOT, which we call glutamate oxaloacetate transaminase enzyme. Why GOT? Because it gets its amino group from glutamate, transfers it to oxaloacetate, and it is a transaminase enzyme. That's why it is called glutamate oxaloacetate transaminase. It can also be called aspartate transaminase because in the event it forms aspartate. Now, the aspartate we have taken into the cytoplasm gets converted into oxaloacetate back. Remember, in the mitochondria, it was being converted into oxaloacetate. So in the cytoplasm, we get aspartate and convert it into oxaloacetate. And in this case, it is a deamination reaction. We are now removing the amine group on aspartate to form oxaloacetate. And guess which molecule accepts this amino group? It is none other than alpha-ketoglutarate. Now, alpha-ketoglutarate, of course, when it gets this amino group, it will get converted back to glutamate. So it is a form of a balancing reaction where we keep on forming molecules on the other side of the mitochondria and inside it, so that we keep on balancing the molecules. And that is very true because every time we have alpha-ketoglutarate formed in the mitochondria moving out into the cytoplasm, that's the time when we have malate entering. And this transporter you see in purple here, it is a co-transporter that moves alpha-ketoglutarate out for every time we move malate inside. And the same thing happens for glutamate. Glutamate formed outside in the cytosol is always entering the mitochondria at the same time when aspartate is moving out into the cytoplasm via this pink transporter. So these two basically move in the opposite direction, purposely to balance the molecules that are involved in this shuttle system in the mitochondria and at the same time inside the mitochondria. Let's look at a very important clinical condition called lactic acidosis. Lactic acidosis is a condition where we have lactic acid accumulating in blood or in cells because we are having rapid anaerobic respiration. We're having the overconversion of pyruvate into lactate. And because this occurs in anaerobic conditions, one of the most obvious causes is hypoxia, where we have a low concentration of oxygen present in the body, and we shall have a crippling of the electron transport chain, so we cannot utilize NADH molecules. And in this case, these NADH molecules are being channeled to be used to reduce pyruvate into lactate to make a lot of lactic acid. So these conditions are very common also in alcoholics. Why? Because alcoholics usually suffer from thiamine or vitamin B1 deficiency, which cripples or slows this enzyme pyruvate dehydrogenase. But remember, this enzyme is very important for making acetyl-CoA. That acetyl-CoA can enter and then be used in the Krebs cycle. And if the Krebs cycle cannot proceed, that means we are basically going to use much of the pyruvate to form lactate because it is not being utilized to form acetyl-CoA. And this is very, very dangerous. Now, thiamine deficiency can also affect alpha-ketoglutarate because this alpha-ketoglutarate dehydrogenase enzyme, which is in the TCA cycle, also utilizes thiamine pyrophosphate as its cofactor. Thanks for watching. I hope you found this video helpful, and I hope the fate of pyruvate made a lot of sense to you. Don't forget to like this video, and most importantly, share it with your friends. Good luck in your next test, and see you in the next video.